Hello everyone, I'm Vinita.
Hi, I'm Shumon.
And today with us we have Dr. Olivier Pourquié. He is Frank Burr Mallory Professor of Pathology and a professor in the Department of Genetics at Harvard Medical School and Professor of Pathology at Brigham and Women's Hospital. He was the Director of Institute for Genetics and Molecular and Cellular Biology, IGBMC in France, and before that, a Harvard Hughes Medical Institution investigator at Stowers Institute for Medical Research in Kansas City. He graduated as an engineer in France and trained with Nicole Douarin. He was the editor-in-chief of journal Developmental Development from 2009 to 2018.
Dr. Olivier authored more than 100 peer-reviewed publications. He's an elected member of the European Molecular Biology Organization and of the Academia Europea. His work on the segmentation clock that controls the periodicity of vertebrae was recognized as one of the milestones in developmental biology of the 20th century by Nature magazine. In 2020, he was elected as president of the Society of Developmental Biology and as a member of the National Academy of Sciences. So with us today we have Dr. Olivier, and you can tell us a little bit about your work, and we are so glad to have you with us.
Yeah, thank you. It's a pleasure to be with you. So the work in my lab has been mostly focused on the development of a particular tissue, which is called the paraxial mesoderm, and which is the tissue which gives rise to the embryonic somites that are the segments of the vertebrate embryo, which give rise to the skeletal muscle, the vertebral column, the dermis of the back, and a few more derivatives. And so for the past 25 years or so, we've been studying various aspects of the development of this tissue, like segmentation. For instance, we identified this segmentation clock that controls the periodicity of somite production. We've been also interested in the formation of the tissue through gastrulation and how this process is linked to the elongation, the formation of the body axis, and how this couples to the activation of Hox genes. We've been also interested in the bilateral symmetry, which is a characteristic of most animals, and how this is implemented and maintained during development. And more recently, in the past 10 years, we've been interested in trying to transpose our understanding of the embryo development in vitro using mouse and human embryonic stem cells. And so we've been recapitulating aspects of the patterning of this tissue in a dish, so recapitulating the oscillations of the segmentation clock and differentiating cells to various lineages such as skeletal muscle or brown fat, in order to also develop more translational applications.
Amazing.
It's very interesting.
So now we are going right away to the questions. Our first question is that when did you started falling in science? Any particular incident that you remember, or the phase of your life when you knew that, oh, now I want to do science, I like it?
So I knew I wanted to do science since I was very young. I mean, it was something I always wanted to do. My dad was a scientist, a microbiologist, and he used to take me to the lab when I was a kid. I don't remember what age, but I still remember going to this lab and the petri dishes and stuff like that. And he was bringing chemicals at home, and I was doing experiments when I was stupid experiments, but they were fun at the time. So that's and then during my studies, I kind of lost a bit interest, and then it came back very strongly. So I decided really that was the thing I wanted to do.
So I just wanted to ask you that, do you remember any of the first experiments that you did?
You mean experiments like manipulated through experiments?
Yeah.
Yeah. In fact, it's not really, well, it is experiments. So when I started I ended up in Nicole Douarin's lab working with François Guillemot, who was a PhD student at the lab. And we were working on was an immunological question, trying to clone the genes of the chicken MHC. And at the time, sequencing was still quite complicated. It was not a done deal, and so when you to get sequence was pretty magical. And so that were the first experiments I did. And also because I had this training in engineering, I was also trained somehow in informatics. So I was one of the only one in the lab able to analyze the data that was generated. So one of the things that really probably one of my first memory as a scientist was to identify the sequence of the class 1 genes from the chicken and realize that they were not where we would expect. And so that was a really strong feeling, like, you feel that you've accomplished something new. So, and though that was not super important, but that was great.
That's amazing.
So when you were doing your engineering, was it engineering and bioinformatics?
No, it's called the National Institute for Agronomy, the school I did, and which in France, it's an engineering school which has some math, some physics, but also some biology. So it was kind of a mix. And but it's historical that it's called agronomy, whereas in reality now it's not that many people end up in doing agronomy. A lot of them do biology or other things.
Yeah.
So when did you realize that you wanted to work on development?
Yeah, initially I wanted to do ethnobotanics. I really like plants, and I'm very interested in the plants. But then I did an internship in Amazonia in Peru, and I realized that the conditions were pretty harsh. The field conditions were. And then when I came back, it was when I was studying at the engineering school. I heard the conferences of François Jacob, who got the Nobel Prize for the lactoseopron. Yeah. And he was lecturing about the discovery of the Hughes genes that were just at this time. Yeah. And so and this was it's called the Collège de France, which is it's an institution where this professor, who are very famous scientists, deliver lectures that are open to the public. And so I was going to these lectures to educate myself, and I was really fascinated by this Hughes business. I have to say I did not understand everything, but it really got me hooked. And then I came back and said, I'd really like to find an internship in a developmental biology lab, and that's how I ended up in Nicole Douarin's lab.
Very fascinating story.
So coming to the field developmental biology, what are the new and exciting areas that are relatively unexplored that you think can be explored a lot?
Well, I mean, of course, one of the very significant development of the past few years is the capacity to analyze things at the single cell level. Yeah. And so that's really changing the way, you can understand this biology, because you can really now go down to the transcriptome level, but at the single cell level. And so, and which means that maybe we'll start to, it start to be possible to understand, things like fate decisions and differentiation at the molecular level. And so I think that's a great challenge for the next year. Of course, there are many more. Another obvious one is the intrusion of physics in developmental biology, and the fact that you have to, take into account the mechanical properties of the cells and the tissues and factor this into the analysis of the embryo. And until now, the vision has been mostly gene-centric, people have been reasoning in terms of gene regulatory network, and I think now we're able to considerably broaden our scope and integrate physics like biomechanics, but also metabolism, which is something we're very interested in and which is very poorly understood, and this actually plays a much more active role than housekeeping. And trying to also imaging, I guess, is also one of the next frontiers, like trying to understand how things happen in real time. And because eventually, there are some very basic features of the body plan organization which are not understood, like bilateral symmetry, for instance. Is why are embryos bilaterally symmetrical? How this gets maintained? And the big questions are still present. Why, Hughes, we were talking about Hughes genes. Why do you have collinearity? All this thing. I mean, a lot of the major questions remain. So I think there's still a lot of...
I feel that spatial collinearity have given direction to a lot of questions that were like just there in the field, and spatial collinearity came and it kind of gave a more focused direction to those questions.
Mm-hmm.
So if you were me and if you were starting your PhD, like I just started my PhD 5 months back. So if you were me and you were starting a PhD, what would be the 3 questions that you would like to address?
Wow. That's tricky, because when you start a PhD, usually you don't know. I mean, it happens more by chance than anything else. You end up in a lab. It's not because I've always been willing to study. So in my case, was chicken embryos and development of B cells was the start of my PhD. I just landed there because people, offered me a position, and that's where I could go. I didn't have a broad question in mind. This came a bit after. I think I don't know if there are some really big questions or small questions. What is important is to have a question, a biological question, and so there are so many systems, so many possible things that one can do in developmental biology that is very hard to. But I would say that the way, at least I like to think about PhDs is first to define a biological question. So in general, that means some descriptive aspects, and then try to understand, the mechanism that governs this particular developmental aspect.
So your lab works in a lot of different questions, and if you would have went to your lab for a PhD, then which one would you have selected?
Oh,
like which question did like you as a new PhD comer would have thought that, oh, I like this, I want to do this?
Nowadays, I'm very much fascinated with the role of metabolism in aspects of the control of development. And for instance, I had a very talented PhD student who I managed to convince to study what controls the developmental rate and compare mouse versus human using the in vitro system of clock oscillations that we developed. And so she did a really nice work and showing that metabolism actually energy metabolism plays an important role in controlling these aspects, which are very fundamental in development, because as you know, I mean, human develops very slowly compared to a mouse, and all this happens at the same temperature. So it's not it's more than a physical problem. But it has very broad implications, like heterochrony, for instance. We know that embryos are able to accelerate or speed up the development of different organs, and so it's a very interesting and poorly explored.
Sounds very challenging too.
So when I went to the fly lab, so the lab assistant there was explaining me, and he said, the moment we entered the incubator room, he said, you keep the flies in 18, they grow in 15 days. You keep it in 25, they grow in 10 days. Keep it in 32, and they grow in, you get more time. You can take a weekend. And I'm like, what? How is that? And I asked him that, how does that happen? Because I kept asking him a lot of questions, because that was my first exposure to a very big lab where like people discussed epigenetics, and like the lab worked in epigenetics and chromatin modeling and developmental biology and Hughes genes. Like there was so much to discuss. I kept asking him questions throughout, like when he was giving me tour. And then he said, if any of us would have known, then we would have been somewhere else. So like, don't ask me that question, ask other students.
No, but it's a great question. I think it's
the
other thing which I find really fascinating is the self-organization capacity of the embryonic stem cells. This is amazing for a developmental biologist. If we could understand how cells and for instance, now we're able to reconstitute paraxial mesoderm, making somites sequentially, and this is completely amazing to me.
So my next question to you is, as a pioneer in segmentation clock, what are the most fascinating aspects of segmentation clock, and how does our current understanding of segmentation clock stands so far?
So I think, one of the fascinating aspects is the fact that, this is like it's a very striking patterning process that's using an oscillator, and that uses this kind of physical object in order to generate a periodic pattern. I think conceptually it's pretty fascinating, and this was predicted by a classical clock and wavefront model in the 70s based on very theoretical grounds. So now what is still very interesting, I think, in this system is that we know that there are oscillations of notch, there is oscillations of wind targets, of FGF targets. We don't understand how they crosstalk, and in reality, we don't really understand the pacemaker of the oscillator. So that's something we're still quite understand in figuring out. Now that we have in vitro system, it's much easier to dissect. So we have real-time reporters to track the oscillations, and in vitro systems where we have populations of synchronized cells. So it's much easier to really get down to the molecular details. And for example, one of the things that we found, which I think is really exciting, is that so oscillations are visualized with a notch reporter, but if you dissociate the cells and culture them in isolation, provided that you inhibit YAP signaling, they will oscillate. So the notch reporter will oscillate, and it will oscillate with the right period, so the species-specific period. And so that really questions the model for notch activation, the kind of canonical model where notch is cleaved, the intracellular domain goes to the nucleus. Because in addition, in these cells where the notch reporter oscillates, so the reporter is lunatic fringe, is a classical notch target, the intracellular domain is not bound to the promoter of lunatic fringe, which means that it works in a different way, and that opens an interesting question, trying to because often a lot of the and that's another aspect of developmental biology which is interesting, is so there are a few signaling pathways that are deployed, always the same, notch, wind, FGF, and so on. But in reality, people don't understand how they work. They say this is notch signaling, this is a downstream of notch. You have no idea what notch is doing. I mean, because it activates a few targets, which are very often the same in all the tissues. And so how it controls the downstream properties is not understood. And so the clock is an interesting system to unravel these properties of signaling, I think. And it also, I mean, it leads to many different questions. So people doing theory, for instance, are very excited because it's an oscillator. It's a very well-defined biological oscillator. So I have a former trainee, Alex Olela, who's been moving very much into this question, trying to look at questions such as entrainment and moving more and more into theoretical issues, which is, a natural direction if you're interested in oscillator. And then there's, of course, the biology, what's downstream of the clock. And there's lots of things that need to be done on the topic.
Okay.
It is very exciting, actually.
Yeah, it's very cool. So coming from science to a bit of life and science, did you had any, like, when students see you, or like when I was reading about you, we always assume that it's so cool, he's doing such a cool science, and you're a supervisor. But did you had any major experimental failures, and how did you manage to overcome that?
Yeah, I had a big one in. So when it was just after my PhD. I spent a lot of time at the time. So I was still in Nicole Douarin's lab, and I spent a lot of time trying to analyze the effect of notochord grafts and various tissue grafts on the development of the somite-derived structures. And so I've done many grafts and found that when you graft the notochord, you can change that triggers homeotic transformation along the vertebral column of the animals. And these were pretty heavy experiments because you have to graft, to do the microsurgery to graft the notochord, and then you have to age the embryo old enough such that they form the skeleton. So and eventually so I felt this was really interesting, that the notochord, was somehow producing something that was controlling the regulation of Hox genes and so on. And in the end, I realized that when you graft the hair, or even if you just open the egg, it's sufficient to trigger. So I had these tons of embryos grafted, and would argue that I might have started with this control, but that took a while, and that was a failure. I mean, I survived it, but it was definitely I put a lot of hopes in this project, and
also that's a lot of hard work.
It didn't go anywhere.
I see it with me and like a lot of my colleagues that we often try to often tend to take experiments very personally. Like, oh, my experiments failed and nothing is worth. So how do you manage to overcome that feeling, if you ever got it?
Yeah, well, one way to deal with this is to try to multitask and have several things cooking at the same time. And this is obviously easier as a PI than as a PhD student. But as a PhD student, it's often a good idea to have, at least 2 projects, not necessarily put the same energy in them, but at least something that if nothing works in one project, at least you get some satisfaction from the other one. I think
So the project that gives pleasure for shorter durations, but it's worth.
Exactly. And and there are some projects that are not risky, basically describing something or looking at embryos. It's always pleasant. So, and it's
and like somehow somewhere that this will work. So
yeah.
So one of the things that a lot of people have a very difficult time managing is striking a balance between their work and life. And as a PI at Harvard, I mean, you have I'm pretty sure you're extremely busy and you have so many things to manage. And so how do you find that balance between your work and life, and how does a regular day in your life look like?
Yeah. So I mean, it changed different periods of my life. The period where, so I've always worked a lot. So I mean, I've been always working on weekends, for instance, because I mean, we're a bit like farmers. It's when you deal with cells or animals, you have to feed them every day, and it's not that so that and also, I felt that if other people are working also on weekends in the lab, I mean, that's part of, you have to be there. You have to be kind of leading by example, because in our work, it's not like in math or in theoretical physics, it's your productivity is linked to the number of hours you spend at the bench. And if you want to get new results, produce new stuff, so you have to do hard work. That's just the way our science is. That said, I think it's very important to preserve your personal life, and I mean, I try to spend as much time as I can with my family, and it's everybody's choice, and for me, the going on weekends or working extra hours, I don't consider my work as work. I mean, it's just things I have to do. So it doesn't really cost me. I mean, when I was director of this big institute in France, that was different because I had a lot of administrative work, and then it was becoming quite overwhelming and not necessarily the most interesting stuff you want to do. But when you do science, I mean, it shouldn't cost you to do that, I think.
Yeah, right. But what do you do when you take a break from science, if you ever take one?
Yeah, no, I do take breaks from science. I don't know. I mean, I like to travel. I like the sea very much. These days, I like to go out fishing. It's very relaxing. Yeah, taking time with friends and family. It's my family is abroad, so I like to go back there and see them. And
so you have been in science for a very long time now.
And
which was the most challenging phase of your career?
Yeah, so I guess that was the when I took over the direction of this big institute in France. It was almost 800 people institute, and then I faced some really really bad politics. So and I was really working very hard, and I think things were working very well, but people disagreed with some of my decisions and strategies and stuff, and they started to really give me a very hard time. And that was the first time I was really confronted with this, a bit like the real politics, like government politics, where you realize that this is a really nasty world.
Yeah.
And so there can be some politics in science, but it's not at this level. You can do your stuff, and if you're successful, and even if you do good science, people will appreciate it and they won't give you a hard time. It's not the same in politics. So that was a tough time, yeah. And so then I decided to move back to the States and to this position at Harvard Medical School and the Brigham, and I really enjoy it. I mean, I much prefer actually having my lab like this and spending time with students and postdocs and little administration. In my case, it was really like you were describing, like, you know, coming back home to try to give back to the country, you know, where I was educated, and so it was pretty painful to be able, you know, to come to the decision that's not going to work out, and I have to move back abroad, and but at some point, you know, it's you have to take tough decisions, and I don't regret it. So it was a very good decision to leave.
So what right now in today's society, one of the most impactful platforms that we see is the social media, and it has a very it has a lot of impact on science and on how science is being communicated today. So what are your views on impact of social media in today's science?
So I mean, I use Twitter quite a bit. I use Twitter mostly as a source of information, you know, people tweeting about their papers and things like that, or meetings. I myself don't tweet so much, except when we have papers from the lab, or sometimes I retweet. I mean, I was brought to Twitter when I was editor-in-chief at Development. You know, I had to have a Twitter account. I don't do Facebook or these kind of things. I mean, I don't know. I mean, that's I don't feel I need it, and I find it a bit dangerous to have, you know, my private life
out there.
Because these things don't go away, and so for Twitter, I think it's or something equivalent. It's a great instrument for science because it's a way to communicate very fast, and I like the kind of organic organization of, you know, your network that progressively builds. It's kind of self-organization, like a developing organism. I think it would be better if it were run by, you know, research organization. I don't see why it's not the case, because the risk is that we are building a great tool, and that maybe one day, you know, Elon Musk will buy it and decide that it's not going to work the way it used to, and then this is all go. So if it were run by Hughes or NIH or something like that, I think that would be much better. But currently, it's a useful tool.
So when do you select a student for your lab? What do you see in them? I
mean, I like the creativity, obviously. I mean, maybe it's I like also the risk-taking aspect, you know, especially in the beginning of your PhD. When you get started, you can think about being a bit ambitious and thinking out of the box. So I like this aspect. I also I think one quality of students is that's, or at least a successful PhD is usually a very mutual interaction. Yeah. So, you know, you contribute to the student development, but the student also contributes to your development, and it should be like you know, it's not like you. I mean, it's great when when there is a true discussion. I think that's the right word.
Yeah.
That you know, that in the end you don't know who had the idea, you don't remember, it doesn't matter, but you discuss, and then you come up, you discuss plans, and I think that's great. So
through the years, you have got mentored during your starting phase, and you have mentored a lot of people. So what do you think is the mantra for mentorship?
To me, it's so I don't micromanage people. So I usually I try to give directions and to correct directions, and also I respect. So some students, for instance, need more attention than others, and I think it's good to respect the independence. In the end, a PhD or even a postdoc is something personal. So it's not. I mean, students or postdocs are not your technicians. You want to make sure that they develop their own thing, and that they get the real rewards of science for themselves, so that they actually find things. So I think that's best when you manage to achieve this. I mean, you can contribute at different levels to this, but that should be the goal.
Right.
Like for instance, I know that some PIs, for instance, would basically tell their students what to do almost on a daily basis, and I think it's a bit. In the beginning, you have to accompany, but.
In today's time, like being a scientist or opening a lab is extremely challenging because the competitions are really, really high, and so what what are the challenges of pursuing to be a scientist according to you in the current structure of academia, and what are the things that one should consider during their PhD and postdoctoral if they want to open a lab of their own?
Well, I guess the most important thing is what you achieve during your PhD and your postdoc, your productivity. And that's, I mean, it doesn't need to be a lot, but it can be important things. I mean, you don't need to have like a lot of papers. You could have like one single, very creative, very original. It's certainly something that would help a lot. And that's actually the single most important thing I believe is that. And so in some cases, I would say in most cases, it translates in some certain kinds of publications, which, like in top tier journal, but not always. And there are some cases, where people have published outstanding stuff in not top tier journal and got very good faculty positions. But in the current system, the reality is that in the majority of cases, the CVs are evaluated based on publications. At least the first pass is done on publications. But clearly it's the type of your achievements which will be the most important. Yeah. I think the rest is very.
Yeah. So as a student, when I started, and I had a lot of problems in reading papers because a lot of them were just not downloadable. And I would mail different professors from different places and then their PhD students to get those papers downloaded because obviously I couldn't afford it. So what is your opinion on open science publication?
Well, I was very big proponent of the preprints when this started. So, as much, and I was the editor-in-chief of a journal, which was at the paywall and all that. But still, I feel this is the way to go. I mean, it's been working like this in physics for a long time. Yeah. And it should be this way in our field. We put pretty much all our papers on bioRxiv. I think I have 18 preprints and that's slowly the landscape is changing. So you see now people are discussing, they're really reading the preprints and discussing. And so that's very important, because for students and postdocs, often it takes 1 or 2 years to publish a paper. Yeah. And if you interview and you have a preprint, it makes a big difference compared to not having this. Exactly. And the preprint is neutral, it's not labeled Nature or eLife or something. So it's just, you have to judge the work. Yeah. So I think it's a very...
Preprints are just before branding.
Yeah, exactly.
So yeah.
But we'll see how the so I've been in the editing world for a bit, and it's a crazy system. The fact that you have to pay this paywall. To me, the best system is really to have the preprints and then publish after reviewing into archival journals like society journals, and but all the hype of the top tier journals is just a money machine. So I don't think it's serving.
We all know that the applications of basic science is extremely important, and to actually see its effect is quite a deal. And so, what are the ways by which translational research can be made more effective?
So my take on this, I've been involved in companies. So I've started 2 companies, biotechs. My personal position is that this kind of work doesn't belong to my lab, but I'm interested in it. So the best way to get this move forward is to try to develop it in an industrial context, and the best vehicle for this is a biotech company. So that's some people try to develop things in there. My lab is a very basic research lab, so testing drugs wouldn't make sense, and so.
Also, with a lot of people now, we see that there's a concept of having some entrepreneurial work along with research. So, do you have any take on that?
I started my first company. So I was a co-founder of a company called Trophos when I was in Marseille, in France. That was when I started my first lab, and and it was a very interesting experience, because what I like and things have changed a bit since then, but at the time, the different fields were very separated. And what I really liked in the company, so we were doing high throughput screening for drugs that prevent motor neuron death, was that we were putting together a team of people, including chemists, engineers, biologists, and we had a goal, which was to develop a platform to allow screening on motor neurons, and all these people had to work together towards this goal. And in academia at the time, these kind of projects could not work, because we didn't have the resource, we didn't have the expertise. It would have been hard to go to a physics lab and ask an engineer to help building the platform, then the chemist to provide. Not impossible, but very hard. Now I would say it's much easier, and probably thanks to the industry. Because a lot of people now realize that you need to be more multidisciplinary.
Yeah, right.
That's true.
So if considering just once that if you have a superpower and you can change one thing in academia, what it would be?
No more grants. Writing.
Wow. Also, since you talked about grants, I wanted to ask this question. I think I almost asked everybody this question that, so when I work, I feel that I'm using taxpayers' money. And that's why I should be more cautious when I work. I should just not shuffle around chemicals, and I should think wisely. So how much do you feel that it is important for laymen to know, to make science more easily understandable to laymen?
No, I think that's definitely true, and it's true that we're using taxpayer money, but on the other hand, we cure cancer and which are layman problems. So
Yeah.
so there is a return on investment, definitely. So I don't think you should feel too guilty.
We always we definitely were passionate about what we are doing, but at times sometimes we feel that maybe this is not meant. I mean, we feel lack of interest, or we feel like doing something else which takes our priority. So did you ever think of leaving science?
No, no, never.
Wow.
No, I mean, I started. So I studied as an engineer, so I was exposed a bit to different other
Yes.
types of jobs, and I saw my friends going different directions, and I did internships in different types of work, and no, I never I feel this is really what works best for me. So I never asked me this question, which means it's the right thing, I suppose.
That's right.
Since you gave a clear no, I would really like to know that you ever felt the most known syndrome right now, the imposter syndrome. Like,
yeah, this is I think everybody goes through this, and you feel, and science is so huge that you feel that you don't understand, I don't know, physics, or and that you should, and then you see people, your friends, who are very good at it, and you're like, oh, really stupid, and why can I not know these things? No, it's absolutely true. Yeah. And I think there are many different profiles of scientists. In fact, I mean, some people are much more analytical. They're very good at mastering everything, all the details and the logic. Some people are more intuitive. They're going more, after the big picture, and then there's many different styles.
Yeah.
So yeah. So and if you're one style, you look at the other style and you say, oh, it should be like that.
So have you ever come across any common misconceptions about scientists?
Misconception, I don't think so, but lack of conception. So people have no real, as you were saying, no real idea of what we're doing and why we're useful.
Yeah.
Often
Scientists are not very well treated, and
so one of the things that is very challenging in both academia and specifically in PhD is to make sure that you are productive and your productivity is at its highest. So what are the strategies according to you that one can use to maximize productivity?
one is to work hard. That's there's no secret, unfortunately. But it's to work hard and work a lot. But as I said, to me at least, it never cost me anything. I never had the impression to sacrifice my personal life, because there was no question that this was what I had to do. So I think it's research is you have to be passionate if you want to be successful, and if you're passionate about your work, then it doesn't cost you to do it. And then of course there are all kinds of strategies. I mean, some people are more efficient, more organized, but I would say that to me the most important things are really to have like the passion, the drive, and the passion, and takes time. I mean, it's just because we have to build the tools, we have to playing with organisms, so this takes time and work. So
yeah. So we are almost in the end of the interview. What's the one best and the worst advice you ever got?
I guess one of the best is I don't remember exactly how this was framed, but was from my advisor, my PhD advisor, Nicole Douarin, was a very famous developmental biologist in France, and who basically stay focused on science. I mean, science is what matters eventually. So, and I think that's true. I mean, if you compared to politics or all kinds of other things, meetings, it's what matters is your science. That's the point. Worst one, I don't know. To be honest, I can't really think about worse advice. Yeah.
That's all right. So, for all the young audience that are listening to you, that will be listening to you, so what would be the one advice that you would like to tell all your young audiences?
I think if you, for people, if really you're convinced that this is for you and you're having fun and it's I mean, it has to be, it's almost vital, it's like, that's why I was saying you don't question yourself about why am I doing this work. It's just that there's just no other option. I mean, I mean, if you're fortunate to have the conditions that allow you to do this, because maybe in some cases it might be difficult, but I think you should not really. It's so another aspect of the mentor comment I was saying is not so much think about your career, but more about how the best science and having most fun and yeah, it's but there are plenty of other respectable jobs where I've had a lot of trainees who ended up being much happier outside of science. And I think that's why I'm saying, if you realize that it's not the job for you, there's no problem looking outside, and there are plenty of opportunities, and it can also make you miserable if you're not for this job, because it's really tough. I mean, but it can also be great.
Yeah. So now we are in the last question of this interview. What do you think about Kitchen Lab?
That's interesting. That was fun. Thanks. I mean, it's a fun exercise, and I don't do this very often, so it's great. And if it can,
for doing it for us.
Thank you so much.
Yeah. Thank you. No, I was going to say, if it can help, young people or trainees, or or clarify things for them or whatever, it's great.
Thank you so much.